deficiencies in homologous recombination repair DNA pathways lead to dependencies on alternative mechanisms. And these can lead to clinical sensitivity to PARP inhibitors or platinum chemotherapies. Currently, there are multiple ways to test for HRD. These including testing for causes of homologous recombination, including mutations in BRCA1 and BRCA2, or for consequences of HRD, including HRD scarring signatures, such as genome-wide loss of heterozygosity. There are currently multiple FDA approvals for the treatment of ovarian cancer, including gene biomarkers of BRCA1 and BRCA2, and for scar-based biomarkers of HRD, including genome-wide LOH or GLOH. We've seen in the last few years building clinical evidence for the utility of PARP inhibitors outside of ovarian cancer, including approvals for gene-based biomarkers of HRD in breast, in pancreatic and prostate cancer. And we've seen that this has led to pretty impressive responses in these phase 3 clinical trials and in the clinic. However, scar-based biomarkers are currently only utilized in ovarian cancer. Based on these PARP inhibitor trials, we believe that there's a greater population that could benefit, ones where we don't currently see HRD with genes only. Therefore, the development of an HRD scarring signature across multiple tumor types is warranted. The use of genomics to identify potential PARP inhibitor sensitivity has been transformative for patient care, with impressive responses across multiple tumor types. We can accurately measure the genotype of tumors using our comprehensive genomic profiling tests. However, we know that gene alterations alone don't capture the entire population, since non-genomic mechanisms exist to induce HRD, such as BRCA promoter hypermethylation. We wanted to build a signature that could detect HRD caused by other mechanisms by examining the phenotype or effect of DNA repair pathway defects across multiple tumor types. Inspired by the successes in ovarian cancer, we developed a novel HRD biomarker to identify the characteristic scars of HRD across tumor types. We utilized a machine learning algorithm trained using our greater than 600,000 patient genomic data set to understand what HRD scarring looks like. We evaluated hundreds of potential features to select those most associated with HRD. And with this, we were able to identify key copy number and short variant features that identified HRD scarring. HRD may be a biomarker for many classes of therapies, including platinum-based chemotherapies, PARP inhibitors, next-generation DDR inhibitors, including selective PARP1 inhibitors and ATR inhibitors. At this year's AACR meeting, we presented data that patients with ovarian or prostate cancer who have an HRD signature are likely to have superior outcomes on PARP inhibitors, and we're excited to expand upon these results at this year's ESMO annual meeting. We're really excited about this because a scar-based biomarker has the ability to select the right patients for treatment across tumor types beyond what the current mutation calling approaches can do. We are building an HRD solution that can be integrated into our validated assay. We looked at patients in the Flatiron Health and Foundation Medicine's joint clinicogenomic database and looked at real-world outcomes on first-line chemotherapy. We wanted to test the hypothesis that HRD-positive patients may benefit from platinum-containing chemotherapy regimen called FOLFIRINOX relative to gemcitabine and paclitaxel. HRD-positive patients had significantly extended survival when administered FOLFIRINOX, with a median overall survival of 15 months versus 6 months on gemcitabine and paclitaxel, with 25% of HRD-positive patients still alive at 2 years on FOLFIRINOX versus only 2% on gemcitabine and paclitaxel. Interestingly, we found that HRD-negative patients had minimal benefit on FOLFIRINOX relative to gemcitabine and paclitaxel, with just a 6 versus 5 month median overall survival benefit. So this study is our first look at HRD-Sig in lung cancer. While Lung cancer is not typically thought of as an HRD-driven cancer. We identified a 4.5% frequency with the highest rates in squamous cell carcinoma at 6.7%. We found that HRD was mutually exclusive with NCCN drivers, suggesting that this is a unique subtype that doesn't have many targeted therapy options. So really a population with need. Our poster includes early data showing an association of HRD-Sig with platinum response. We are very interested in expanding this cohort and looking at additional tumor types as well.